Hello everybody, I am Chandan Singh. Today we will discuss about the topic of ribosomes. That is very important topic and it will come in the many medical entrance exam like NEET as well as other than competition exams. So now that is very important topic for the 11th class students. So firstly we will discuss about ribosomes structure as well as other than things. So just focus on the board please. तो यहाँ पर मैं बात करूंगा फ्रेंड्स कि फर्स्टली व्हाट इज द डिस्कवरी is given by that particular scientist, that is who is discovered, that is Pallet, the scientist is discovered by the ribosomes. Exactly, ribosomes is also called factory of the protein. So that is also known as protein factory of cells. That is very important to know about every student. Okay, how can we learn about the factory of cells? That is also known as by the ribosomes. So ribosomes is also known as the factory of the cells. Now second thing is ribosomes is the particle which are occur in the inside the cells, and that is very important cell organelles which do some different type of activities. Like firstly, it is absent like this. It is absent in the cell of RBCs of the mammals. It is absent in the cell of the RBCs of the mammals who one is having mammary glands and doesn't have any membraneless organelles. These are called membraneless organelles. Now this one is the property of the ribosomes. Now the next topic, what is the size of the ribosomes? Size. So it is approximately 15 to 20 nanometers is size. These particles of the ribosomes is very important for the purpose of the protein synthesis. Why is required protein synthesis? Because all the living organisms have their body functions is depend on their protein structure, because we all are made up of the proteins. Our body organizations about 90% is made up of proteins, and proteins must be required to each and every living organism. So protein synthesis must be required, and who will do to perform protein synthesis? Ribosomes protein synthesis is play the role. Now it has two subunits. First one is large unit and second one is a small unit. These both the unit are depend on their types. So basically I am talking about the next step of the type of the ribosomes. So basically ribosomes is two type, 70S and 80S. So 70S having also two subunit, 50S as well as 30S, and 80S two subunit, that is 60S as well as 40S. You doesn't count like this ke 50S and 30S having the total number of quantities 80S. You can't count like this. You just measure this unit like this. 50S is the large unit and 30S is the small unit. These both units are connect with the MGIs. MGIs is the most requirable element enzymes which having to responsible to bind to subunit. If the concentration of the ribosome is a minimum 0.0 molar quantity is must be required to bind these two units. If the concentration of the molar quantity of the amines is less than, so both the unit are separate to each other, and the quantity of the concentration of amines is more than 0.001 molar, so it will connect with each other. Both the units are interconnect with each other. So that is very important to know about the every student, कि how can be two subunit of the ribosomes are interconnect with each other. So how can we elaborate this concept? Ribosomes basically is two subunits of two parts. Basically, first part of the ribosomes is 70S type, and second one is the 80S. Both the units having two subunits. 80S having 60 and 40, and 70S as 50 and 30. Both the units are linked with each other with the responsible of the enzymes. If the concentration of the enzyme is less than 0.001 molar quantities, so it will separate to each other. If the concentration of the enzymes 0.001 is more than concentrations, so it will connect with each other. So that is responsible. The connectivity is binding to connectivity between the two subunits is responsible by the only enzymes concentration. Now second one is point. The next point is, whenever we are talking about this polysomes, what do you understand as polysomes? Sometimes ribosomes, these are ribosomes, that is smallest particles, are bind on mRNA. Messenger RNA, that is the part of the ribosomes. RNA. That is the part of the RNA. That is messenger RNA. It will bind with the mRNA and form the what? Form the a chain. ये chain को forms करता है. It will form the chains that is called polyribosomes. It is also called polysomes. It will also called ergosomes. These are the some names. You just note down in your notebooks. If you having the question arising in your exams, what do you understand polysomes? What do you understand ribosomes and ergosomes? So you just denote in your mind whenever the ribosomes particle are interconnect or bind on the mRNA chain of the RNA, so it will form a chain. So what is required to binding the ribosomes particle on RNA? Enzyme. Enzyme is very important to binding the ribosomes on mRNA and form the bond. It will form the bond that is called peptide bond. Peptide bond form the chain that is called polyribosomes. Polysomes are called ergosomes. So that is the chain is why is required. Second question. This is the chain why is required, because human body or any living body. You can't you just take the example any living organism. So I just take the example human body. Human body is formed 90% formed by the protein. Human body is composed 90% part of the protein. So how it is formed? First one is basically the cell of the human being having nucleus, and nucleus having the genetic material that is called DNA, deoxyribose nucleic acid. It will further break into the RNA, that is a single part of the DNA. single strand RNA, that is very important. So this process is called transcriptions. Now it will further break into the protein. So who is responsible? Ribosomes for the breakdown of the protein, but this process is called translations. So that is the very important process, transcription and translation. It will do the next step for the protein synthesis and form the body organization of all the living organisms. So you just note down the things, okay, what is required for the protein synthesis. So who is responsible? That is ribosome must be required. That is very important cell organelles inside the cell, as well as it will also be found in the mitochondria and chloroplast. Now, in mitochondria, ribosomes is also called oxysomes. And in mitochondria it will be counted as the M1 particles. And whenever it will form the chain on mRNA, it will form the chain that is called polysomes, polysomes and ergosomes. This is the chain is very important for the purpose of the protein synthesis. So it will require for the protein purpose, because protein is a highly requirable element, most abundant element for the formation of the body organization of all living organisms as well as human being. So human body is formed via the proteins, because it will 90% part of the protein. So human body as well as all the living organisms to form their body organization with the respect of the protein. So protein is must be required, and it will form by the ribosomes. It is non-membranous structures, and it will not occur in the RBCs of the mammalian cells. Fine. So this is the little bit informations about the ribosomes. One thing is very important. Ribosomes is denoted by the 70S and 80S. So what is the meaning of the S? S is the Swartberg unit. That is the sedimentation unit, that whenever we have to take the a pot and some particles of the ribosomes are centrifuged by the different rate. So the particles which are sediment on the rate of the 40S, so it will counted as the 40S, and the particles which are overlap on the surface of the pot, and that is counted as the 30S. So this is the differentiate the different unit, 40S and 30S, 50S and 60S, whatever we have to count it as the centrifugation process. So that will the rate of the frequency which are counted of the ribosomes particle on their sedimentations. So that is very important. That is the sedimentation as Swartberg unit which are required, which are denote the centrifugation process, centrifugation's rate of the ribosomes. So that is denote the naming of the ribosomes. Okay, what is the rate it will sediment are on the bottom as well as the surface of the pot. So that is the very important rate of the particle of the ribosomes, which form the two different unit as well as 80S as well as 70S. तो यहाँ पर मैं आपको यही बताऊंगा 70S और 80S जो हैं वो दो अलग अलग यूनिट हैं जो राइबोसोम्स को अलग अलग रेट पर सेडिमेंट करती हैं। जो बड़े रेट पर सेडिमेंट हो रही हैं वो सरफेस पे बॉटम पे चली जाती हैं, और जो लाइट पार्टिकल्स हैं वो सरफेस पे आ जाते हैं। तो दोनों को अलग अलग काउंट कर लिया जाता है, एंड इट विल काउंटेड एज डिफरेंट यूनिट 70S एज वेल एज 80S। तो फ्रेंड्स, ये आपको कैसा लगा? तो यू जस्ट गिव द कमेंट। फिर से नेक्स्ट वीडियो में मिलेंगे, तब तक के लिए बाय बाय, थैंक यू।